The overall goal of this protocol is to describe how to direct colloidal self-assembly by anchoring supramolecular moieties onto the colloids whose interactions are strong, directional, and reversible.
This method can help to answer key questions in the material science field, such as how to accurately control the colloidal self-assembly to create complex and mesostructured materials with interesting properties.
The main advantage of this technique is that colloidal association is driven by the supramolecular moieties that remain responsive to light and to temperature upon surface immobilization. Therefore, colloids self-assemble upon photoactivation of the hydrogen bonds of the molecules by UV light.
To synthesize the fluorescent silica seeds, first mix 2.5 milliliters of difunctionalized aptes with 25 milliliters of 25% ammonia and 250 milliliters of ethanol in a 1-liter round-bottom flask.
To obtain monodispersed silica seeds, it is really important to add the tetraethyl orthosilicate under the meniscus of the reaction mixture.
Using a glass pipette, add 10 milliliters of Teos while stirring with a magnetic stir. Similarly, after 5 hours, add another 1.75 milliliters of Teos and stir the mixture overnight under an argon atmosphere. The following day, wash the seeds as described in the text protocol. To synthesize the core shell silica particles, first add ethanol, deionized water, 25% ammonia, and the seed dispersion to a 1-liter round-bottom flask. Then, fill a plastic syringe with 5 milliliters of Teos and 10 milliliters of ethanol. Fill a second plastic syringe with 1.34 milliliters.